If you come to work with me, yeah, you're gonna get a lot of pizza. We just refurbished our balcony, so we're gonna get an arsal, so also. Bon, anyway. So I'm just gonna steal a few minutes of your time because last time I didn't finish this. We were talking about, and I started to talk extremely fast. So just to be, close this corner. We were talking about how to localize sounds in space, and we're talking about two Qs, ILDs, level differences, and ITDs, interval time differences. So the times come in different times to the far ear and then to the near ear first, and then the far ear. And this is computed in a nucleus called the MSO, the medial superior olive, and from there it goes to the inferior colliculus. So there's actually, this is nice and well known, and you can see this in the textbook. And the idea, before we even recorded from these, people recorded from these regions, the idea was that this can be computed by coincidence detector. This is called the Jeffreys model. And the idea is that there are delay lines based on axons, actually the cables of the axons. So if input comes here, it will first hit the first neuron, etc., etc. And because there is a delay between the ears, let's say sounds come from the right-hand side, it will first hit the right cochlea ear and then start, the input's going to start to travel, but then it's going to hit the far ear, but here it's going to be the first delay station, okay? It's going to arrive earlier in this first in the hierarchy of the delay line here, and the coincidence detector is going to be there. Basically, if you record from this red neuron, it will spike only in a given ITD, the difference between the right and the left input, and we can say that we can construct these receptive fields, or ITD, responsiveness of each neurons, and they will not normally lie on the zero time axis, okay? We will have neurons that will have a zero ITD, but normally you can see shifts in the ITD. This is at the level of microseconds, as we saw in the last slide, because in the earlier slides of this talk, because of the size of the head, okay? So in humans it can go up to 600 microseconds, in small animals and birds it can go to normally between 40 to 200 microseconds. That's the range of ITDs, plus minus. Okay, so this was the model, and of course if the sound comes from the front, this will be the coincidence detector, and the neuron will spike at ITD 0 in this case. There's a nice animation that you can see on the web. Okay, anyway, you get the idea. This is just a depiction of this, so I can go fast. It was just an example. I don't want to waste too much of our time. But basically, if you record from neurons in the MSO, this is an example from the cat, one does record these ITDs, okay? So you see that the neuron spikes with the highest firing rate at a given ITD. But because of phase ambiguity, because the neuron doesn't know if it's the first part of the phase or the second part of the phase, these ITDs you can see the responsiveness of the neuron even if the shift is at the period of the frequency of the sound. Okay, so if this is a 1,000 hertz sound, a 1 kilohertz sound, there is going to be it's going to peak at the ITD. In this case, it doesn't really matter. I don't know, 50 or whatever, the specific neuron. But if you look at the period of the 1 kilohertz, it's going to be 1 millisecond. So therefore, it's going to be, you're going to see replications of this tuning curves along the period of the frequency. Okay. Now, this is called phase ambiguity because the brain doesn't know if it's 50 microseconds or 1,050 microseconds. Okay, the decoder of this system doesn't know. So one has to figure out or solve this phase ambiguity. The thing is that the idea is that given ITDs are here regardless of the frequency tuning of the cells. The cells also have frequency tuning, and each neuron will show say, the same ITD. This is a given neuron recorded at different intensities. You can see that it's tuned best to 250 hertz and has an ITD, in this case, of 600 microseconds. Okay, so you see that because it's tuned to 250 hertz, if we go beyond the best frequency of the cell, we will get generally less and less activation on one hand. However, the ITD of this cell is the same at all different frequencies. So it will peak here around 0 at 600 microseconds. But if you look at the different frequencies, because of the sound frequency, the period is going to be different. And if you remember that we talked about how to construct a click, right? Remember, we have to put them all in the same phase, but if you add more and more and more frequencies, you're going to end up with a perfect click, and all the rest is going to be summed up and cancel each other, and the average of these are going to be 0 eventually. If you put all the frequencies in the click, this is how the next station actually disambiguates the phase ambiguity, because it sees neurons of ITD from all different frequencies. And they all are synchronized here, but are dispersed away in this regime beyond the 600 microseconds. Okay? So that's an important point for the phase ambiguity. This is an important issue. Okay, so but the basic idea, again, from Jeffries, and now we have recordings from cats and gerbils, etc., is that there is these coincidence detectors that in essence form a place code. So if I'm coming beyond this issue of phase ambiguity, if I'm looking at this map, I know that this is the neurons here. I have 0 ITD, meaning that the sound will come up front. Okay, so this is essentially a computed map of space location, unlike vision and somatosensation, which is a physical map, so to speak. Okay, the neurons themselves are sensitive to a given region in the environment. Here it is computed from the left and right side. Okay, this is just sums up all I said in the last lesson. Essentially, okay, that neurons from the cochlear nucleus send excitatory information into the MSO. These are called EE neurons, excitatory neurons, because they get 2 excitatory inputs, and only when these 2 coincide, this is a coincidence detectors, they will spike. The ILDs are computed, and then from there it goes to the inferior colliculus, the next station. The LSO, where the level difference is computed, are IE neurons, inhibition excitation neurons, because they get excitation and inhibition through this huge synapse. You remember this monster synapse, which is highly sensitive. So basically the neurons here get excitatory and inhibitory input, and they send inputs to the contralateral inferior colliculus, and this gives the inferior colliculus the ability or the character to respond to contralateral sounds more pronounced, and this is a very general theme of the brain, you know, that it crosses to the other side. So in the cochlea, of course, we will hear ipsilateral sounds, but at the inferior colliculus already, all the sounds that we hear have a preferred contralateral sequence. Yes?
So you talked about both of these being in are in coincidence or coincide.
What do you mean? In the MSO,
Yeah.
neurons are coincidence detectors.
Like in time.
In time,
Right.
at the resolution of microseconds. Okay,
Right.
because of their phase locking characteristics.
Right. So what's the time? I mean, it's hard to believe it's exactly the same time, right? So it has to be in some kind of time window.
What do you mean?
I mean,
The relevant ITDs, because of the size of the head, it can go from 0 if the sound comes from the front, ITD 0, perfect, or in the back, of course. And the maximum is 90 degrees. For humans, it's 600 microseconds. For birds, it's small birds, I don't know, 40 or something microseconds. And the animals that normally people test, the barn owl, etc., it's around 200 microseconds. So between 0 and 200 microseconds, that's the only information we can use. Otherwise, we're going to have
no, but I mean,
that's the cue.
you have
That's
no.
the range of the
What's
cube.
the time
Right.
window of integration?
No, I mean,
Microseconds.
Right. So
what?
how small differences
Yeah.
can we actually detect?
Right.
Yeah, so this was the first,
Right.
I see, the perceptual resolution. This was here, okay? We can detect down to 1 degree in space, humans, which is about 10 to 15 microseconds in ITDs, and about 0 or up to 1, around 1 dB in ILDs, which is quite remarkable. Okay? Anyway, but so in the textbook stuff is this coincidence detector of Jeffreys. That's what you will see in all textbooks. But, however, and this is really proven nicely, the Jeffreys model has proven nicely in the barn owl. And the barn owl, although it's an amazing model, we have to realize it's a unique model. It's a very special animal. Okay, it's a binaural specialist. It hunts only based on at night, only based on hearing. It actually uses high frequency sounds to hunt, and that's a very uh, unique phenomena. Its neurons in the equivalent of the MSO can phase lock at these frequencies, which again is quite remarkable. You remember that our neurons can phase lock up to 2 kilohertz or so. And yeah, and you can see what?
Bats do the same thing.
Bats are also very special. Yes, but they are very special animals. The question is whether we use, or all other mammals use the same. This is not a mammal, but for the bat, all other mammals use the same story. In bats, it's even more fancy story because they use echolocation, they emit sounds, and they look at its reflection. We won't have time to talk about that. But the basic notion, you look in any textbook, this is what you find. Jeffrey's model, and the proof of it is the barn owl. However, you must just realize that people have very, very strong arguments against this now in mammals, that they think a barn owl is a special case, but it's not true in mammals. And there are several reasons for this. For one thing, if you look at physiological ranges of ITDs, okay, I cannot get information if the ITD will be 700 because it's not physiological, it's out of the range. So one has to look in the range of ITDs which are physiologically relevant to the specific animal. And what one finds is that the range of ITDs, if this is the full range, in mammals is either out of the range, normally out of the range completely. So it doesn't make sense that the ITDs of the neurons are really computing something physiological. That's one problem. The other thing is that we can phase lock only in very low frequencies. And when you look at low frequencies, because of the properties of the slow frequency, it's more sluggish in time. So, the wave is wider. The sinus is wider when it goes down, and it covers the whole physiological range. In the barn owl, you get about 3 pulses in the near ear before it reaches the far ear. Okay, this creates a problem of phase ambiguity, which is solved in the way that we talked about before. So this is one thing. Okay, that these receptor feeds are sluggish, and it didn't make complete sense of how can we solve it with these kinds of ITD machines. Okay, so when you look in the Gerbil specifically, it is unlikely that more than one cycle will reach the near ear because we're doing it in low frequencies. And the ITDs are very broad compared to the barn owl. And people were recording from, and that's one thing. And then there was a big Nature paper in 2002 showing a critical role for inhibition in the MSO. So I told you that the coincidence detector is an EE neuron. It's getting input from the cochlear nucleus here and here, whatever the delay lines, it doesn't matter. But it's an EE neuron and a perfect coincidence detector. It appears there is a strong role for inhibition, and if you shut down inhibition, all the neurons lose their ITD tuning and fall onto the 0 spot. Very big paper in Nature, highly cited, very influential in this field. Therefore, they said, guys, we cannot neglect this anymore. The ITDs, this is the physiological range of ITDs in the gerbil, in this case, I think it's a gerbil, okay, that they recorded. And this is the peak of the ITD. You see, it's completely out of the physiological range. So something doesn't add up. So now there is a big force of scientists Benedict Grote, David McAlpine from UK, and Benedict Grote from Munich, which are pushing very hard that the textbooks that we teach the students are wrong in mammals. So the barn owl is a beautiful story, but it's a very unique story of this Jeffreys model. And our data, we see the data, and it has to explain it nicely. It's probably not the same thing. And I was planning to do a discussion about this. If you look at these kinds of curves, if you look carefully, you can look at it in a different way. Not by the peak of the ITD, but what you can see that in the physiological range, all the neurons have the most sensitive region of the curve in the physiological range. So basically, all the neurons, and this is just different frequencies, are indeed sensitive within the relevant physiological ITD range. You see that every small shifts in the ITD, and that's the whole thing that we can actually detect, okay, physiologically relevant for us. You see that in all the neurons are tuned around the region of the physiological ITDs in their peak of their dynamic range. This means that very small changes here will be reflected in very high changes in the firing rate of the neurons. Okay? So now we're trying to look at this thing not completely as a place code, but rather as a rate code of the changes of the neurons. And now there's big fights between the people. They'll say it's a place code, and there's a perfect map, and I can just say where the neuron is and detect where it is on space. And the mammalian people, this is the guinea pig, sorry, say that we actually use a rate code of this information. And not only that, we cannot. The computation is actually done in the inferior colliculus, which is the next step up, which has to compare two different rate codes. Okay, so if the stimulus from the location goes left, then there's going to be a decrease in one ITD hemispheric information and an increase in the other. And this is the actual comparison, and not the coincidence detector. Okay, so people spend their whole careers, on fighting this, because the notion is that you give the spoon to the students at all levels is this coincidence detectors of Jeffreys, but I just want to say that there are reservations to this model, and it's important that you realize these reservations. Yes?
In the previous slide, the maximum is only like slightly out of the physiological.
The maximum of the higher frequency neurons,
Yeah.
okay?
So
Actually, perceptually, we are much better at these lower frequencies, okay? But still, these are frequencies that can phase lock, think up to 2 kilohertz.
the black curve is the lower frequency?
The black curves is the lower frequency, yes. You see, by nature, because of the phase locking and the sinusoid, they're normally more sluggish. But we would expect that they would all phase lock on the same spot. There's also issues that the slower the frequencies, the higher the ITDs. But in this case, surely, we can't use 1 millisecond or 1.5 millisecond apart. Doesn't make sense. Okay? Like tell you one, yeah, shoot.
Is like all of the axons were roughly the same size?
No. So you will read this in the textbook, okay? Some axons from the contralateral side have to make up for the speed, so they're better isolated, speed is higher, and actually some of the axons from the ipsilateral side are more sluggish, they're larger, and it takes more time to get, okay? So it's an important point, but it's not completely critical. It's just more evidence, anatomical evidence, supporting the Jeffreys model in the barn owl, especially. Okay? In the barn owl, both the anatomy and the physiology fits very, very well with the Jeffreys model. Okay, just 2 more slides. One is that now all the information that is coming from all these brainstem regions converge in the inferior colliculus. And this basically is where the inferior colliculus can now take the information from the ITD, from the ILD, from the spectral cues, which we didn't talk about, and Lilach will talk about a little bit now, and do the integration. Okay? But most of the problem is already solved here. That said, if you screw up the auditory cortex, there's going to be all kinds of messes in a little bit ITD, because there's a lot of reinnervation into the colliculus. So one thing is that many cues converge in the inferior colliculus. Unfortunately, I do not have time, but this inferior colliculus sends also a copy of information to the superior colliculus, where information from the retina, the visual map of the world, is overlaid on top of the auditory map of the world. And there's a beautiful story of this convergence. Okay, it has to do with eye movements, and the reference is actually the direction of the eyes. Okay. Anyway, it's a beautiful story also about plasticity. Unfortunately, we are not in this course able to cover this topic. And yeah, so basically this is the superior colliculus where it merges. I just copied this from the book, sorry for the low quality. But if you look in the superior colliculus, you will find neurons tuned perfectly to a spot in space after all the computations of ITD, ILD, etc. have been done. Okay? So this sums up what we talked about, and you remember that unfortunately again the order is a little bit screwed up because of unimportant reasons. Okay, that we're doing now the talks, and only on Sunday we're going to hear about auditory cortex. But we talked about the inner ear, the cochlear nucleus a little bit, the nuclei in the brainstem that compute spatial information. From here, information goes to inferior colliculus, the thalamus, and then auditory cortex. On Sunday, Elin Elkin will give us 2 hours about the auditory cortex, his view of the auditory cortex. And basically, with that, we will complete the auditory system, of course, after today's lecture. And then we're going to have another series of 3 talks and cake. ובא לציון גואל. After, no, you still have to do an exam. ובא לציון גואל. It's like something with anyway, they will explain to you. Okay, so we are now ready to rock. This is it?
Yeah. No worries, take my. Yeah, just the slideshow itself is long.
And I apologize for not being on
Computer
time.
itself.
Hmm?
The computer itself.
Excuse me, right?
Oh wait. Oh.
I have questions.
Start from the beginning. So this was my talk. Anyone?
What's the cat?
Okay. So it will take a while before we get to the break. I'm going to talk with you about context-specific reweighting of auditory spatial cues following altered experience during development, which we'll break down soon and understand what it means. But first, the main agenda, the researchers, they wanted to see how spatial hearing develop when first experience acoustical input that changes in time. I want to see not just about the auditory system, it's a model question first thing for the sensory system in general. How do they adapt to different environments that get different input that have to adjust to different dynamics? So first we'll have a very quick reminder about sound localization, then we'll get to the main question and what exactly the title means. Then we'll get to how the question was actually assessed and the results. So first, sound localization in 3Ds. We want to find out the azimuth, which is in the horizontal plane, the elevation, which is in the vertical plane, and the distance. And in this study, they focus only on the horizontal localization. So in order to localize sound, we use both binaural and monaural cues. The binaural cues, we talked about them extensively in the class. We have the interaural time differences, which are supposed to be accounted mostly for the lower frequencies, and we have the interaural level differences, which are supposed to be accounted mostly for the high frequencies. Then we have the monaural cues, the monaural spectral cues that we use. This is thanks to the mammal external ear that can filter the high negligible for the horizontal localization in normal conditions. And those cues can actually help to localize sounds only in higher frequencies and only if we have some prior knowledge about the spectral properties of the sound which we are hearing. Otherwise, we have no way to know what is the gain from the monaural cues from the ear and what is the source stimuli itself. So those cues can be described by the directional transfer function. This function explains for each azimuth and each location in space how much the sound is attenuated for those conditions. And it's different among different individuals due to differences in the head and the ear shape, but it's pretty similar, so it can be evaluated. And it can be measured by placing microphones inside the ear, playing sounds, and then compare what we record to what we play. So what happens when people can't hear in one ear? That's why people have a very big problem with localization, mostly in the horizontal plane. And especially if the hearing loss occurs at a very young age, there is some kind of adaptation. So people become much more sensitive to the monaural cues, and they learn to use them, and this improves a lot the way in which they can localize sounds in the horizontal plane if they have some prior knowledge about the sound that they're supposed to be hearing. And the problem with this plasticity is that sometimes it's irreversible. And after normal hearing is restored, there is amblyopia, there is a weakening of the input from the ears that was deprived, and people can't localize sound well even though they have both ears intact. A similar phenomena happens in vision also. Oh, any questions so far? Okay. So in this study, they asked a bit different question. They asked what happens if you don't apply a stable unilateral hearing loss, but only intermittent hear loss. In order to check that, they took ferrets and they raised them with a plug in the left ear, and most of the time they were with the plug, but from time to time they'd allow them a little small breaks of normal hearing, and they did it since the age of hearing onset. So the question is, what will happen in that case? How will the auditory system learn to adapt and learn to localize sound? So they assume there will be some kind of adaptation, that they will learn to use the monaural cues much more than the binaural cues. But what will happen afterwards when we take the plug off and when we actually let them hear with both ears? Will they still use a lot more the monaural cues that are stable, that they learn to count on, or will they in some way learn to acquire different models of the environment, one for each context, and switch between them? And why do we care about this question? So first, for practical reasons, due to ear infections, infants usually have long periods of hear loss, and this can cause amblyopia. So many of the times, if even afterwards, when they restore their normal hearing, they still can't localize sound, they can't distinguish between the sound and the background, and we want to know how to prevent that and how to maybe reverse that. And then there's the bigger picture question of to understand how sensory systems adapt to different environments. So we can go back to the title and see what it actually means. So context-specific reweighting of auditory spatial cues following altered experience during development. So context-specific is just how are we while we're being tested? Do we have normal hearing or only one ear? Weighting of the auditory spatial cues is how much do we use the binaural and the monaural cues, and experience in development is how were the ferrets raised? Did they have normal hearing or not? So what they did, they wanted to actually measure the weightings of different cues in different contexts with different individuals. So they did pretty much three things, and they got them pretty much to the same results in different levels. First, they performed behavioral tests of sign localization. Then they created a model of localization that can help to distinguish between reliance on monaural and binaural cues. And they applied the behavioral results to the model to actually see what the first did. And then they did cortical recording in order to find the reweighting of single neurons in the auditory cortex in different contexts. So first the behavioral test. So they have juvenile plugged ferrets and control ferrets, which were raised with normal hearing. They were tested with or without the earplugs, and they were all shown with 200 trials of 200 milliseconds of the broadband node. It was in one of two conditions, either flat stimuli, which means that for all the frequencies in the stimuli they had pretty much the same attenuation, or the spectral randomization, which means that for each frequency they had different attenuation. And the task was to localize the sound, to say which one of the 12 speakers surrounding them gave the sound. You just need to approach the direction, and they got a prize. So the result, we can see that without the spectral randomization, here we can see that on the x-axis the stimuli location, on the y-axis the response location, and the size of the circle is just how many of the trials were in this place. So we can see also here that with the plugs, the controls did horribly. They couldn't localize sound at all. Without the juvenile plug ferrets, with the plugs did quite well. They did a lot better, which means there was some kind of adaptation here. And without the plugs, they both did very well. There was no difference between them, which means that the ferrets can both adapt to the plug, but adapt to the normal hearing situation and localize sound just fine in those conditions. And with the spectral randomization, the results were a bit different. With the spectral randomization, the source sound is not constant, so we have no way to know anything about the spectral properties of it. In that way, it's impossible to localize sound using one ear. So, as you can see here, the ferrets with the plug could not do it, but when the plug was taken off, they did it just fine, which means that they did in some way use binaural cues in order to do the sound localization. Questions so far? So, next thing they did some computational model of localization that will help distinguish between different strategies. In order to do this, first they needed a way to represent the sound as it comes to the ear. Then they created some kind of a decision-making model for both monaural and binaural listeners. They did it using a MATLAB program. Then, after they had decision-making mechanism, they wanted to restore the mean stimulus that led to its response. This is done using reverse correlation maps. I will explain about it soon. And then they want to compare the obtained reverse correlation maps to the original directional transfer function, which represents the cues from the single ear, and see how similar it is, which means how much they relied on those cues. So first of all, in order to represent the stimuli, he took the source stimuli and they filter it with the directional transfer function for certain azimuth. So they could measure the DTS. They did it with some other ferrets from another experiment, and each time they could pick the azimuth they want. They filter the source stimuli, and then they get the filtered stimuli, which is how the stimuli from this location would sound to the ear. Then they made a decision-making model. For the monaural model, it was simply based on correlation. They had the filtered stimuli and they had the DTS for all the different azimuths, and they just showed the location that maximized the correlation between the original DTS and the filtered stimuli. And this did pretty well for the flat stimuli, but not so well for the randomized stimuli. And here the results pretty much matched the behavioral results. And for binaural listeners, they use the intensity level differences between the filtered sounds to both ears. So afterwards, they wanted to construct can see here is, in red we can see the mean stimuli over all stimuli, and in gray and black we can see the mean stimuli that elicit a 60-degree response. So we have the mean stimuli, the stimuli that elicit some response, and they check the differences between them if they are big enough to be significant. And this gives the reverse correlation map, which says for each frequency and for each azimuth how much gain was there according to the decision making of the model. Now, the next thing they wanted to do is check how similar it is to the DTF, so they checked the correlations between the two, and they found out that for higher frequency, where we can actually find something, there is a very high correlation between the DTF and the reverse correlation model. Here each dot is the frequency and the azimuth, and we can see the gains for the different methods, and we see, what we can see is actually that the decision making can be explained by the DTF, which in that case is pretty trivial because they use the decision making mechanism that's based solely on the DTF. Here is the same reverse correlation map for the binaural model. So this model gives us kind of a proof of concept that we can use reverse correlations in order to find out which cues the listener used in order to localize sound. You can see in the monaural model that we have many gains in the higher frequencies, and in the binaural models we have lower gains and spread across all the frequencies and all the locations. So the next thing we want to do is actually to take the behavioral data obtained from the first experiment and then put it into this model, take the compute the reverse correlation maps and see which one it resembles, which means which cues they used in each situation. So this is what we did, and we see that the results here are pretty similar. For controls that were without the earplugs, we see that we have small gains spread all around, and for the juvenile plug first with the plugs, we see that we have high gains in the higher frequencies, which means that they use the monaural cue. And also for the control, we can see that they use the monaural cues to some extent, a little bit, but a lot less. Then they wanted to see if there is any type of monaural dominance. So again, they took the mean DTF for each ear and the mean RCM for the controls and for the first with the plugs, and they computed the correlation between each RCM to each ear. They wanted to see for each group if the correlation to the right ear is bigger than the correlation to the left ear. So here you can see the differences between the two. And you can see that control used the right ear and the left ear pretty much for the same amount. There is no difference between them. While the plug, the first used the right ear a lot more, which is pretty trivial since they were wearing the plug, so they had only the right ear to account for. So now what would seem reasonable to do is actually to take the parrots that were with plugs and see how they do without them, and then see which cues they use. But for some reason they didn't do it. They just moved on and tested it in a different way. It seems to me like a main drawback of the paper. So the next thing they did cortical recordings of single neurons in the auditory cortex. What they wanted to do is find the single neurons and give each of them a weighting index, the index that will say how much they use the right ear and how much they use the cues, all the other available cues in different conditions. So they recorded from anesthetized and controlled the juvenile plug ferrets. They took neurons with a higher characteristic frequency. Their stimuli was again 100 milliseconds of version noise, but right now it was filtered and played directly into the ear canal so that they can manipulate the sound to each ear separately. And each recording was done under two conditions. One is a normal hearing and one is a left plug to the left ear. This is done just by filtering the sound. So the cues that they checked were the ILD. So in order to test the ILD, they just play different levels of sound to the different ears for 0 degree azimuth, and the azimuth in its ear, and they just played different different degrees into the different ears, and in order to test, for example, what was the effect of a 90 degree in the left ear, they just took all the trials in which they did 90 degrees to the left ear and all the trials from all the other azimuths in the right ear and collapsed them and saw what happened. Which, again, they didn't explain why they can do it. They didn't say why they can play contradicting sounds to different ears and still rely on the monaural cues the same way. I hope they know what you're doing. So, the procedure first, for each unit that was tested and each hearing condition, they calculated the joint distribution between the cue and between the spike count in the response window that followed the stimulant in that trial. So what we can see here is in normal hearing conditions for the left ear virtual azimuth, for each possible azimuth in the left ear, how many of for each possible spike count, how many of the trials made it. And the same for the virtual plug. Is that clear? So, from the joint distribution, they calculated the mutual information between the cue and between the spike count. Now, mutual information is widely used in information theory, and it's a measure for two random variables for how much knowing one of them can reduce the uncertainty about the other. Just the entropy of one variable minus its entropy given the other. So what we are left with is the entropy that can be explained by the other. And what's nice about it is that it can be calculated directly from the joint distribution. So they did that, and they calculated the mutual information for the right ear and the spiking rate, and for each other cue and the spiking rate. So we have MIR, and MI other is just the sum of the mutual information for all the other cues. And from that they constructed the weighting index for that specific neuron, which is just the difference between the mutual information for the right and the others divided by their sum. Which means that the weighting index increases if the neuron relies more on the right ear and less on all the other cues, which is exactly what we wanted to find. So they calculated this weighting index for the different neurons in the different conditions, and what they found is just like they expected and like they found before for the behavioral result is that when those who were raised with the flag they used the cues from the right ear a lot more than all the other, the weighting index was high. But in all other conditions, when the earplug was removed, they used all of them completely to the same amount. There's no difference between the index for the controls and for them. Which means that they can adapt to different contexts and use the they can adapt to different contexts and with the plug use only the monaural cue, but without the plug use the binaural to the maximum amount, extract all they can out of it. And the significant effect was found only in the left hemisphere, which is ipsilateral to the plug, not in the right hemisphere, which can imply that there is a contralateral influence here. Because when they tested the left hemisphere, then they found that with the plug, the neuron used their cues from the right ear, but when they did it in the right, they didn't find any difference. So maybe it wasn't using the cues from the right ear. So, to account for all those results, they describe this as a mechanism of experience-dependent optimal cue integration, which means that the system can adapt to different contexts based on the strategy that it learned to utilize, and every time when we are faced with a task, based on the current context, which is which cues do we have available right now, and based on the experience, which means how much we learn to use each strategy in the past, we choose the optimal weighting among the different cues. And this can also explain the use of spectral cues in horizontal localization, because when we do have binaural cues, we use them, and we use their spectral cues in a very little amount, and when you have no choice, then we use it a lot more. And this is a good news for therapeutic benefit for amblyopia, because we know that system can adapt to different contexts. So if we can maybe play children who suffer from deficit in the hearing, play them sounds and maybe a higher volume, or let them adjust to it, then maybe we can reverse or prevent the effects of the amblyopia. And the last point they made is the ferrets were all tested while they were anesthetized, which means that other higher order mechanisms such as attention can't be needed for this process. And in studies that were performed before, they said that this different cueing might be the result of higher order mechanism, but here they prove that it doesn't rely on that. So this is the method already. Question?
Bon, the floor is open for questions. Any question? Okay.
What about you?
Yeah, you made me read the paper. So
Started to read the paper.
yeah, almost. Yeah. Okay, it's a hardcore paper, huh? Bon, no questions. We can go to the next part.
רק לקחת לי מייק.
לך על זה.
כן, עוד מישהו, משהו? עמידה?
Take a break first.
לא, no break. רק לניר.
Is the order important for you? Because
רגע, הנה, שמתי ללילך שעון והוא עכשיו נכנס.
Just in time, huh?
Okay, the 7 minutes ahead of time. You went fast.
Why? It was more than 20 minutes.
What?
It was more than 20 minutes.
20. Okay.
25 I thought.
No, I put 23, okay. Kama, 20?
yeah.
I'm coming from a different course. 25. Okay, bon.
Okay, so for those who don't know me, my name is Nawaz. I'm going to present a paper from Nature published in 2013 by Znamensky and Zador from the Cold Spring Harbor Laboratory. As you can see, its title is Corticoparietal neuron in auditory cortex drive decisions during auditory discriminations. So as you can see, it relates to the auditory cortex. I hope it'll be a nice primer for this Sunday presentation. I hope I'll get everything right. So the basic question that they were investigating in this paper is where does acoustic information relevant to action selection process? That is, we all experience the world. We all know that sound can give us a lot of information, and one important behavioral consequence is whether to take this route or that route, or where should we look for food, and how should we use and employ sound to achieve these behavioral consequences. More specifically, what they are asking is how do outputs from the auditory cortex establish associations between sounds and motor responses. So if we think about the auditory environment as somewhat arbitrary, that sounds don't really have a predefined meaning, but we have to learn mostly the meaning of most sounds. How do we form these associations between the significance of sound and the motor responses that are appropriate? The possible answer, the hypothesis that they're suggesting and then testing is that projections from the auditory cortex to the striatum carries the acoustic information that drives behavioral choices. So just a bit about a background about our two regions of interest. So as you can see, you can see my pointer, that's good. So this is the auditory cortex. As you see, it's in the end of the route, the auditory pathway that we studied in class. And the auditory cortex has projections to different areas in the brain. That goes to higher regions like the prefrontal cortex, goes to motor areas, goes to emotional or limbic areas, and it also has projections to the striatum. The striatum is part of the basal ganglia actually made up of the putamen and the cauda nucleus. You can see it here. And going back to this figure, you can see this white area. And what we know about the striatum and basal ganglia actually in general is that it's very related to reinforcement learning, to choices between different motor actions and procedures. And we most most findings that we have that relate to the striatum comes from studies that investigate the associations of striatum with the prefrontal cortex, the motor cortex, or limbic regions. There are not, at least according to this paper, not so many studies that investigated the associations of striatum with the auditory cortex. Now, just as you can see in this figure, just a very general outline of informational flow in the basal ganglia. It's a very linear flow. Information is relayed from the cortex. That includes the auditory cortex to the striatum. And then there are two main pathways, the direct and the indirect pathways. I won't go into it. And then from there, there's information going to the thalamus and back to the cortex. And that shows us how this loop does show that it's a very good candidate to a sort of information processing and decisional processes area of interest.
Is the most
Yeah.
the frontal cortex or the acoustic cortex?
The cortex in this figure?
Yes.
The cortex,
The
including the
cortex.
auditory, the motor, everything. Now, really relating to the auditory cortex, what they know that there is a specific area in the striatum that receives projections only from the auditory cortex and not from any other area. And that's why it can be a really good candidate as the pathway that by which acoustic information is drives action. So to better investigate how acoustic information drives motor responses, they used behavioral tasks, auditory discrimination tasks, in which we have a rat in a middle position, and then after some pre-stimulus delay, we have either one of the two different types of tones that we'll get to it in a minute. Each tone, the rat learns to associate it with one of the two cells, either the right or the left. And if he learns the test, he knows that if a certain type of tone appears, he'll get his reward as water in one cell or the other. So they say that the rats were able to very quickly learn this test. They were, of course, on a water diet, so they were quick to learn this. The two types of the tones that appeared were either low frequency tones from 5 to 10 kilohertz or higher frequency tones from 20 to 40 kilohertz. Now, there were not only two types, only two stimuli, but rather the different trials have different, a train of 100 tones presented. It could be either a perfectly only 100 low frequency tones, or 100 high frequency tones, or a mix, a combination. And that way we can do not a discriminant analysis, but more of a continuous estimation of how different stimulus, auditory stimuli is processed and drives action. Now, this figure C, as you can see, for each tone that is high, you get a 1, minus 1 for a tone low, so minus 1 represents a trial in which all 100 tones presented were from the low frequency, and 100 for all 100 tones were presented from the high frequency. And as you can see, that in these extremes, the proportion of the the number of trials in which the rat chose the right cell was almost perfect. This figure is actually important, so if somebody doesn't understand it, we're fine? Okay. So in addition to this accuracy behavioral measurement, we also had a response times of the measurement, electrophysiology using tetrodes, local field potentials, and deriving from that single unit activity, and also the implemented optogenetics, and I will get to it in a minute. Just to show preliminary results, so if you know, the auditory cortex encodes different tones according to their frequency, and each area or each single unit is thought to be tuned to certain frequency. So if you can see here, this is the auditory cortex neurons. You can see the activity before the onset of sound and then after. In the gray areas, before the rat started to move to one of the areas, and what you can see that this cell responds more during the high frequency tones. This figure E is taken from a striated auditory projecting neuron, and you can see that also there is a some tuning curve. On a more average estimate, this black curve shows the entire behavioral consequences. It's just actually like this this figure that I showed you. But the red one is what they refer to as the neurometric curve, meaning that the spiking activity of a single neuron is also differs with the type of stimuli that is presented, and that's obviously just a different way to show this. But what they're trying to show and say is that the two curves are similar. So for
So
the
this PSTH would fall where on the curve? On
the what?
so the PSTH in the raster in A,
Yes.
where will it fall on the right in C? It is like the most extreme case, the maximum high frequency response?
This these are different trials.
I understand, okay, but where you said this is a reflection,
Right.
C is a reflection of A. So
Right.
where would you place A on C?
You mean like in terms of time, the x-axis, like right after the
I
sound stimulation?
Where is A reflected in C?
Here in the red curve,
So,
but
so this same neuron will be along the whole red curve.
yes.
Yeah, because you see also low frequencies and high frequencies.
Right.
I see. And this is one neuron.
Yeah. So for the more interesting results that are obviously involving optogenetics, I know a lot of you already know this, but if somebody didn't take the appropriate courses and I'll be too fast for him, please stop me. This is not the first experiment that they did. Obviously will not address all experiments, but in the second experiment that they did, they used two combinations of molecular techniques that their aim is to express channel rhodopsin only in neurons going from the auditory cortex to the striatum. You know that channel rhodopsin is a channel that excites depolarized cells when you present certain light pulses. To achieve this specificity, what they did, they injected a herpes virus with 3 into the striatum, which also projecting neurons to this area take up. And into the auditory cortex, they injected a virus that expresses both flax and CHR2. And when in a certain neuron you have both the 3 expressing virus and the flax CHR2 expressing virus, you get only in those cells channel rhodopsin expressed. So they did this, and then obviously they used in some of the trials light stimulations, and when you stimulate in those trials that you stimulate, you get a higher activity of those neurons. Now, in figure E, the black curve represents the control, the trials in which they didn't have light, and the blue represents for a certain neuron how its curve changes with stimulation. Now, this specific neuron responds more to low frequency tones. That's why we see a bias that is constantly reduced from the control, because what actually happens when we activate light is as if that rat hears additional low frequency tones, and thus his response is more driven to the low frequency. Is that clear? Yeah. Now, this is for a single neuron, and this is for all the different neurons. What we can see that for those cells preferring high frequency, the bias will be higher, meaning that the blue curve should be above the control. And for the low frequency preferring neurons, we'll have a bias towards lower frequencies. Okay? where would it be?
Yeah, it's on the bottom left
Which exactly
quadrant.
of the dots is it?
Yeah, but it's on the bottom left quadrant, one of those.
Yeah.
It's not the most extreme example that they have.
No. Now, if you can see in this technique, it is possible that the activation of this neuron actually activated another additional recurrent loops inside the auditory cortex. So in order to rule out that possibility, they did additional things. They changed a bit the setup. First of all, instead of stimulating, presenting light in the auditory cortex, they did it in the striatum. And secondly, they induced a pharmacological measure to block all activity in the auditory cortex. So in this setup, what we can do is we actually excite the axon area of the neuron. And in figure I, what you can see that they measure, do electrophysiological measurements in the area of the soma of those neurons. And we know that there isn't a backpropagating action potential. So before we apply pharmacological measures, we do get some voltage. We see voltage changes in the auditory cortex. But when we use some blockage, we see that there is no such activity. And of course, after some time, we restore the backpropagating activity that might be causing additional recurrent loops inside the auditory cortex. Now, doing this technique, what they show that when you, both in the previous experiment and in this experiment, you get an increased bias. It's not significantly different, but it is significantly different from uninjected control rats. So they show that the effect of the auditory cortex on the striatum is not related to recurrent loops inside the auditory cortex, but is specific to the pathway going from the auditory cortex to the striatum. And as you can see, this is again similar to the figure that I showed before. And you see that this is a significant effect going for overall all neurons that they measured. Now, after doing excitation, obviously the next thing to do with using optogenetics is doing the reverse procedure, and that is instead of depolarizing cells, is inactivating them using hyperpolarization, hyperpolarizing receptors. So again, they did the Curie coupled with opsin receptor expressing combination. This time they express ARCH, which is an inhibitory, has an inhibitory effect on those neurons that are activated. And as you can see in figure C, when light was activated, the activity of the neuron was eliminated. And if you think about the behavioral consequences or what the rat might be sensing in that area, if before he was listening to a tone and now you eliminate the delivery of that acoustic sound, that is as if you're making the rat not hear certain tones instead of increasing those tones. And thus, we will see a reverse effect, a reverse bias, meaning that when we do this for low preferring frequency neurons, we'll get actually as if they're preferring more the high frequency tones and vice versa. Okay. So a summary of the results. This one I did not show you. The first thing that they did is they just activated auditory cortex with not the specific usage of the Cree. And when they did that, they had a bias. They did found bias in activation, but it was going to all different directions. Only when they did a specific activation of the cortical striatal neurons, they had the positive bias correlated with tuning, and that was also had with normal performance. And the first experiment they had reduced performance. And the last result that I showed you is that in activating those pathway neurons in that pathway had a negative bias that is correlated with the tuning of each neuron, and was also accompanied with normal performance. According to the researchers, the importance of their study is, first of all, that they characterized auditory striatal neurons during behavior. Before that, they say that it was done only using anesthetized animals or passively animals. Also, they said that this is a study that estimates the contribution of single neurons or single units to behavior during normal perception. This is again from 2013, and we all know that this is a relatively new development. And lastly, they claim to identify a general mechanism for the control of motor decisions by sensory context. However, if we go back to the general question whether indeed the projection from the auditory cortex to the striatal is the only pathway, as they showed, that drives, that helps the animal make decisions, there is some problems with how they designed the experiments. First of all, and this is something that you couldn't pick out, is that optogenetic manipulations, the locations in which they implemented them, varied from one experiment to another. So for some experiments they did it in the striatum, for others in the auditory cortex. They didn't really provide I think, convincing or any reason why they did this once here and that time there. And I would expect them to do it constantly in both regions or just in one region. But more important, if you remember, one of the experiments included the blockage of all or most activity in the auditory cortex, and despite this fact, we still saw a bias and effect of acoustic information on the activity of the behavioral consequences of behavioral choices. And that really surprises me. If you block the activity of the auditory cortex, how did the information reach the striatum? That might imply that the striatum actually does receive additional information from other areas which compensate for this loss. So thank you.
Bon, questions? Nothing.
Clear.
Okay, let's take a break, a 10-minute break. קצת ורבע. No, 1:20. We'll be back.
Lines are yelling at me.
Could the leader take a quick note? So we're going to take a break.
Last presentation. Another one of those papers with a title of 3 lines. So I'm gonna do pretty much the same as Ila. First gonna go a bit into what is really the issue that we're trying to investigate here. So this paper is trying to investigate the issue of functional connectivity. Now, what's functional connectivity? In respect, for example, to the structural connectivity. When we think of the structural organization of the brain, we know these things about layers and columns and stuff like that. So the functional connectivity is overlaid on that structure in the sense that the functional connectivity determines how the information actually feeds through these networks of neurons that are pretty static. I mean, obviously, the structure changes. We know this for like learning and things that they induce slight changes in what the layout of the brain actually is. But the functional connectivity really is about the rapid modulation of these networks so that they can perform the function that they're meant to do. So this paper is trying to do a little bit of new thing in a sense that so far functional connectivity is often researched in terms of just trying to make cross correlations between neurons, or you record a bunch of sites, you see activity in each of them separately, and then you see cross correlations, and from that you try to deduce how these sites are connected. But that type of investigation doesn't really tell us much about the inhibitory activity and how the inhibitory activity shapes the network function. Now, why is inhibitory so important? Well, think about it. If we just have activation of an activation of an activation, like eventually your brain would short circuit. So, it's pretty logical to think about inhibitory activity in the brain as a major component of what shapes the network state, such as the oscillations and up-down states and other determinants of how the information actually flows through the network. Now, an issue with inhibition is that most of the inhibitory neurons that we know about, if we even know about them, they're just kind of difficult to characterize. So there's this one population of inhibitory neurons, the PV plus neurons. We've heard about them before. They're actually quite well characterized, so we can target them because, well, they're PV plus, parvalbumin expressing, and they make up a really large portion of what we think are the inhibitory neurons. So over 50% of them we can target using PV, and they also are functionally have been strongly implicated in this connectivity issue in the sense that they've been shown to provide feed-forward inhibition, feedback inhibition. So we know that they have an important function. So in this paper, we're going to try and see if we can figure out what exactly they do. How do they modulate the network connectivity so that the flow of information becomes different within layers and between layers. So now that we've painted this picture again a little bit, functional connectivity, what is it all about? Let's go back to our paper with its beautiful title of 3 lines. So what are we going to really do here is try and combine the optogenetic tinkering of these PV plus neurons with a few computational models to see if we can indeed deduce the function of the inhibition that comes from these PV plus neurons. Cool. How are we going to do this? So if we want to look across layers and between layers, our recording mode of choice would be this polytrode that we show there. So it's a 4x4 polytrode that they place in such a way that it covers layer 2, 3 through layer 4 across a few columns, hopefully. And we make these recordings in PV3 mice. So we target the inhibitory neurons, the PV neurons, using the double flux, inverted, all that sort of stuff systems. We have expression of channel rhodopsin in about 50% of the PV neurons. So then we use the silicon polytrode to make actual recordings, and the recordings are made while animal listens to sounds, as it were. These are pure tones that are presented just for a very, very short time. And on half the trials, the tone is, as it were, covered with light activation. So if you look here, this picture covers like 5 trials. Each trial lasts only a second, and each tone tip is really just 25 milliseconds, very short. And around the tone in half the trials, we have optogenetic activation of the PV neuron. So really we have inhibition. So later we're going to take these recordings and use these 2 models mainly to look at what we actually did. But first let's take a closer look at the methodology. So they were really quite thorough about this in the sense that before just poking the polytrode, they poked around a little bit with tungsten electrodes to see where exactly A1 is and to identify the tonotopic axis. You may remember it, that corresponds to the apex and the base of the cochlea. So we look at how the tonotopic axis sits in this particular mouse, and then the polytrode is lowered parallel to that axis. So hopefully we'll be able to record from sites that have a specific threshold, a specific bandwidth based on where it is spatially localized. Alright, so then we make our recordings in 16 sites. Only 14 of them actually worked properly. And we calculate a few basic parameters, and these are separately calculated for light on and for light off trials, and also analyzed separately, and then we can compare them later. But these are just really the basic things that we all have seen before. The spike count, the PSPHs, all that sort of stuff. So the threshold for each of the sites and the bandwidth of activation, and the signal to noise ratio, like how much does the stimulus actually determine the response. Alright, so everybody clear on the methods? We know what we're doing? Cool. Now let us turn to the models. First of all, the icing model. Now, my physicist friends here in the class will probably know much better what this is about than me. It's a model that comes from physics and describes coupling between 2 places. So it's different from traditional correlation analysis in a few ways, mainly that it takes into account the site-to-site coupling between all sites. So whereas traditional correlation analysis, you're like sort of just looking at 2 points separately every time, the icing model will recalculate weights for all sites more or less simultaneously. And in that sense, it will become much less prone, for example, to false positive activation, as we will see in a bit. But first, let me try and explain to you a little bit about this model. So it takes as input binary matrices, so just zeros and ones, of the spiking activity and of the stimulus. So spiking activity is represented in 5 millisecond bins. If there was a spike in that time bin, then there's a 1. If there wasn't a spike in that time bin, then there's a 0. Then the stimulus is represented basically in the same way. So if there was stimulus, there's a 1. If there wasn't a stimulus, there's a 0. Except there's 22 different frequencies. So these are each represented separately. Then what the model really does is it takes these 2 inputs and it tries to predict the output, the X, given the stimulus, using matrices that it makes that are sound-to-site coupling and site-to-site coupling. So the weights of these matrices are learned using minimum probability flow learning. That I'm really not going to get into right now. If you want to, there's a link there that explains it pretty nicely. But basically, point being that you have an objective function, you try and minimize the error, and that way you find weights that are most likely to represent well how sites are coupled to each other and how sites are coupled to sound. So how do they respond? And then you can, so the nice thing about all these learning models is you can have them learn on just a fraction of your data, like 80%, 90%, and then test how well your model works on the remaining 10%, which is exactly what they did. They evaluated the log likelihood of the model using cross-validation. So you basically just do this a bunch of times. You take 80% of the data, learn, test on 20%, then take a different 80%, learn, test on 20%, until you've learned from all data and tested on all data. So you get a pretty good estimate of how well your model actually works. Now, in order to also compare the Ising model with traditional cross-correlation analysis, they also took a reduced form. Yeah?
Can I ask a question?
Of course.
So you said that bins were 5 milliseconds in time, and they didn't change, right? I mean,
No,
the
they
bins
were
were, it was,
in all Ising model analysis, they just took time bins of 5 milliseconds.
and the 5 milliseconds is around the time, I mean, at time 0, at 0 milliseconds, this is the time where the tone was, the sound was,
No, sound was
given?
presented in the middle of the trial. Like all these, the light on was like, if a trial was 1 second, then the sound was exactly in the middle for 25 milliseconds, and the light stimulation was for 100 milliseconds, I think, around it. So, I don't know exactly what your question
Okay,
is. But
sorry. So for each light activation, this is at the beginning of the bin? Or, I
no,
mean,
the beginning of the
or-
bin is just
So
whenever the
bins are
trial starts.
just randomly 5
No,
milliseconds?
the bin length was chosen based on like if you see the mean activity and the standard deviation. So based on that, they chose the value for the standard deviation to be less than 3 times mean. But basically, it shaping the window in such a way that you don't get like really weird big activity, but also not really weird small activity.
So they took the entire trial and just
Yeah.
dissected it into 5 milliseconds, and the
Right, 'cause
time
you
windows
can't-
were not changed at all?
No, you can't really calculate with continuous time in these models. That's the only thing. So you have to choose some kind of time bin. A 5 millisecond came out as a good one for them, but it's not really that important. I don't think. I don't know.
I think you must choose a width for your unit of your calculation for the matrices.
Yeah, of course, but I mean, it will change your results drastically if you change the time bin.
Yes.
Right, it might.
Yeah,
But so
if
they
you
were
take it
pretty
too large,
precise
they're
about.
all gonna be positive, right?
Yeah.
There's gonna be activity
Right.
in all of them. So you have to, it can't be less than 1 millisecond. And it can't be larger than, I don't know, 20 milliseconds.
So I guess my question is it, I mean, what's the biological correlate of 5 milliseconds?
You can
Yeah, they-
fit in 5 spikes maximum, even not that, normally 2 or 3. And I think it depends on the brain region also. And the cortex is pretty sparse, so 5 is you're capturing most of
Right.
the activity.
It is actually one of the strange things I think about this study, because later we'll talk about vector autoregression, in which they took a time bin of only 2 milliseconds, which is less than half of 5. So I'm not 100% clear on how and why they chose this, but it seemed to work for them. So
In
I'll
our
just go with it.
analysis, we take 10 millisecond bin because the firing is very sparse.
Right. Anyway, so back to the Ising model. So it comes in 2 flavors. We had the fully connected Ising model, as I just explained it to you, where we have sight-to-sight and sound-to-sight coupling. And then we have the reduced Ising model, which they call the independent neurons model, in which they only look at sound-to-sight coupling. So that resembles more the traditional correlation analysis. As you can also see here, here they correlated how well the correlation analysis correlates with the independent neurons model, and the correlation analysis with the fully connected model. And you see that the independent neurons model is actually really well correlated with correlation analysis. They're pretty similar. Whereas the fully connected Ising model really is less correlated. So it really does something different from the traditional correlation analysis. Now, does it actually work better? So if you look at the log likelihood of the independent neurons model versus the fully connected model, we see that the fully connected model indeed performs better. It's more predictive of the activity than the reduced model. So indeed, these sight-to-sight couplings, they add to what we know in our analysis. And then what we see here is that using the fully connected model, we can indeed recover connectivity that is qualitatively different than what we would see in correlation analysis or using the reduced model, in the sense that In this picture, we see increased activity mainly in the bottom two layers, row 4 and row 3, which probably correspond to the thalamorecipient layers. So, knowing what we know about brain anatomy, this seems to make sense because those are the layers that receive the direct input from the thalamus and should be more responsive to stimulation. Whereas here, what we see in layer 1 and 2 as activity is probably just false positives due to the way correlation analysis works. And then, another cool thing that this Ising model does is you can actually see this structure that we know about, the columns and the layers, emerge from how the analysis works. So, if you look here at the correlation in a layer, they're much stronger than correlation in columns. So then this is just during regular being basically. So we see that there's really much more connectivity within a layer than would be in a column. Whereas in the site-to-site correlation, so the traditional analysis, there's basically correlation more or less everywhere, and you don't really see this spatial structure emerge.
That's the G,
What are you?
so the Eiling is the left and the...
This is Ising, fully connected, and this is site-to-site correlation.
Okay.
So with site-to-site correlation, we see this structure much less clearly, if at all. Whereas the Ising model seems to discover it pretty well, even though it doesn't directly get any input of the spatial layout of these recording sites. It just tests all the correlations. Alright, so are we convinced that the Ising model is pretty cool? Alright, fine.
Coolness factor, that's the most important thing.
Right? So now let's see what it can tell us actually about, what PV plus neuron activation does. Well, first of all, what they did is to plot the bias term of the Ising model, which they took as a measure of firing activity overall, basically. So really what we are looking for here is just to see that in each of the cases where you turn on the light, overall firing activity goes down. That makes sense, right? You turn on the inhibition, you see less firing activity. Fine. And these differences are all significant. They're bigger here in row 4, and really small here in row 1, but all of them are significant.
What's the y-axis now?
So the y-axis is the bias term.
Bias, what?
The bias term of the Ising model. So they took it as a measure here of the overall firing rate. I don't know why they didn't just take overall firing rate, but whatever. But then here in B, it's a similar picture, except here we see the site-to-site coupling for columns and for rows. And what's interesting to see here is that when you look within layers, we don't see any real effect of turning on the inhibitory activity. Whereas in columns, we see an increase of functional connectivity. Is that clear?
Where is the increase?
So here, we see that light on actually changes the coupling between sites within columns, whereas within layers, turning on the light doesn't really do much.
This is a paired analysis, or?
So this is the Ising model.
So you have to put the whole thing in.
Yeah. All of this is just a whole... I guess it's a drawback, but at the same time an advantage. You can really look at the whole population at the same time. Alright, so I think that's a pretty cool result. And then to look at that a little bit more closely, again, we see these thalamorecipient layers, the bottom ones, 3 and 4, that really gain much in connectivity activity when the light is turned on. So these are 3 representative examples, where in light off condition you see that there is not much correlation, and in light on condition the coupling increases much for these bottom 2 layers especially. Alright, so from that, it seems we can conclude that turning on inhibition, or turning on these PV plus neurons, enhances functional connectivity, especially within columns, but not so much within layers. Well, one problem with this analysis is that we can't see which direction these changes go. So this could be feed-forward connectivity, it could be feedback connectivity, it could be both, it could be neither. And in order to take a closer look at that, we used vector autoregression.
That's your 20 minutes.
Oh, really?
Yeah.
Shit. Are you guys bored already, or can I go on for a little bit? So
No?
I'll try to be fast. Basically, this again is a model that learns, except instead of taking just one time point, it takes the population activity of previous time points, so up to 40 milliseconds, and uses the whole population in order to try and predict the activity of a site. And then you do some fancy stuff with output nonlinearity and whatnot, but basically it's a linear model in which weights are assigned to each point, and the weights can be used to measure, again, the functional connectivity. So what we see here is, like in any case, basically the same layer, if you try to predict activity of a layer of neurons, the layer itself will be the best predictor of how the activity, what the activity looks like in that layer. But in these cases, activating the PV plus neurons doesn't really change the activity. So that's not really interesting to look at. What is interesting to see is, again, layer 4, turning the light on increases its influence on the other layers. So if we're trying to predict the response of any row, then in light on conditions, the activity of row 4 is more predictive than in light off conditions. So from that, again, the layer 4 is a cellular recipient layer, and from anatomy we know that, we get first input to layer 4 and that feeds to the other layers. So it seems like we can conclude that the connectivity is in feed-forward direction rather than any other direction. Never just believe what you see. We're going to test it a little bit more. There's a few findings here that are maybe a little puzzling, maybe a little unexplained mostly. But first of all, they answered the possible criticism that the connectivity increase that we see is because of